Well, good morning, Biology 30s. I am going to continue talking about meiosis. So we talked about the importance of meiosis, and one of the things that we said in our last video was that the DNA has to make a copy of itself ahead of time before meiosis occurs, and this is happening in the interphase, the S phase, just before meiosis 1 occurs. And this is to ensure that each of the daughter cells is going to have enough DNA so that each of the daughter cells will be able to perform and function normally. So a couple of key events that we're going to be talking about. This video is focusing on genetic diversity. And so meiosis, we're creating gametes and we're creating offspring that are not the same as the parent. They're diverse, they have variety. And so the first major event is occurring in prophase 1, and that event is called crossing over. The second major event that's going to increase diversity, that's going to scramble the genes to make offspring different than the parents, is happening in metaphase 1, and it is called independent assortment. So with prophase, in crossing over, the tetrads are coming close together, and the maternal homologue and the paternal homologue, so like the chromosome from mom and the chromosome from dad, are getting nice and snugly and close together, and they call this synapsis. This is when crossing over can then occur. This is kind of like the event leading up to it. And so you can see that the non-sister chromatids, so like one of mom's and one of dad's, is kind of intertwining and combining. And what happens is a little piece breaks off and there's a swap. There's an exchange of genetic material. And that is what crossing over is. So that this chromosome does not go unchanged throughout this meiosis process. It is changing, right? It's swapping, it's exchanging genetic material between the paternal and maternal homologues. So that's crossing over. Then the random way in which the tetrads line up on the metaphase plate is going to be another way to scramble the DNA. So you can see here's mom, the maternal chromosome of this pair, and then here is dad's, the paternal chromosome. And so that means that when it undergoes lining up here along the metaphase plate, then when it gets into telophase, this tetrad is going to separate into two chromosomes. Okay, so the pair, the homologous pair of chromosomes separates, and now mom's chromosome goes to this pole, dad's chromosome goes to this pole. We see that's the exact opposite for this tetrad. There's dad, the paternal, will go to that pole with mom's, and here is mom's going to that pole with dad. So we see that we're scrambling up the genetic material based on, let's say that this forms the egg cell that is going to be fertilized by a sperm. Well, it's going to have some of mom's genetic information, some of dad's genetic information, and it's totally going to be different than this cell that forms. If this is the egg cell, then instead that is fertilized by a particular sperm. Okay, so this independent assortment is the random way that these tetrads are lining up in metaphase down the metaphase plate. So meiosis 1, this first division, we're separating the tetrads, the homologous pairs, into chromosomes. In our second phase, we have meiosis 2, which is pretty much just the same as mitosis. We are taking chromosomes and making them into chromatids. So, I mean, the names of the phases are all the same: prophase, metaphase, telophase, but there's no important events in this stage in meiosis 2, the second set of divisions. No important stages here for genetic diversity. So when we look at the steps of meiosis, remember interphase is occurring before meiosis 1, and the S phase is when the chromosomes are replicating, they're making a copy. And that first division, prophase, metaphase, anaphase, telophase 1, that's our reduction division. We're separating the tetrads, the homologous pairs, into chromosomes. Then there's an interphase, and I want you just to think of it like an intermission. There's no DNA replication between these, okay? You can't have the chromosomes replicating or you would be right back to the same number of chromosomes you had at meiosis 1, and then that just defeats the entire purpose of you going through meiosis 1. So this second division is just like mitosis, and we're now separating the chromosomes into sister chromatids. And again, there's no important events that are occurring in meiosis 2 for diversity. All those important events happened in meiosis 1. So again, you should practice drawing and labeling the stages out of meiosis, knowing the major important events that are occurring and which stages are important for genetic diversity. And so that's our prophase 1, where our sister chromatids are coming together. Our tetrads are coming together during synapsis and crossing over. So at each point where crossing over has occurred, we have a little chiasmata forming. And the crossover is the swapping, exchanging segments between the paternal and maternal homologs, and that is resulting in scrambled genetic information. And then during metaphase 2, depending on which way the chromosomes line up, like is it maternal going to this pole, and then here's the maternal one here going to this pole. So maybe if it flipped around, we'd have more similar to mom, or if both were dad's chromosomes pointing to the same pole. So it's just random the way that the tetrads line up during the metaphase 1. Okay, so both those events are important in increasing diversity. Here is our meiosis 2, separating out chromosomes into sister chromatids, same as mitosis, just that we're ending up with 4 cells. Each are going to be haploid. Okay, so just a reminder, meiosis 1, reduction division. Our tetrads are becoming chromosomes, right? 2N to N, and they're still duplicated. Important stages, prophase 1, where crossing over occurs, and important stages, metaphase 1, where crossing over occurs. Anaphase, we're just taking those homologous pairs, and they are now separating into chromosomes, and they're moving to opposite poles. Our meiosis 2 is same as mitosis. Here we have our chromosomes. They are still duplicated, but we've already undergone our reduction division. So they are haploid N, but they are still in a pair, right? 2 sister chromatids make up 1 chromosome. So it's duplicated, and it is going to form a chromatid, which is each of them are N, and they are unduplicated. Okay, so same as mitosis, we've got 2 different cells here, and in each of them, here's the chromosomes on the equator, and the chromosomes are then separating into chromatids. They move to opposite poles, and then at the end of meiosis, what do we have? We have 4 separate cells, each are haploid, and we have just made some gametes for reproduction, for sexual reproduction. Okay, I wanted to show you guys a couple of quick videos here. The first one I have queued up for you is McGraw-Hill's unique features of meiosis. So let's watch and listen.
Although mitosis and meiosis have much in common, meiosis has 3 unique features: synapsis, homologous recombination, and reduction division. Following chromosomes.
So it's actually missing one important characteristic there, and that's independent assortment. And I'm going to watch that on our next video on random orientation. So let's continue on here.
Replication. Homologous chromosomes, or homologs, pair all along their length. The process of forming these complexes of homologs is called synapsis. The second unique feature of meiosis is that genetic exchange occurs between the homologous chromosomes while they are physically joined. The exchange.
So here's this exchange of genetic information. This is crossing over. So the chromosomes came close together, the tetrads formed, and then the maternal and paternal swapped. The very ends there broke off, and now we have chromosomes that are not identical to what we started with. And of course, that is crossing over. That's huge for genetic diversity.
The process that occurs between paired chromosomes is called crossing over. The third unique feature of meiosis is a reduction division. At the end of meiosis, each cell contains only half the original complement of chromosomes. Because of the
Oops,
crossing.
I'm just going to rewind that for a minute. I'm
Have
just
the
going to go
original complement
back,
of
stop there. Okay, so what we see here is independent assortment occurring. Here's our, that was a terrible straight line. It's not a straight line at all, but you can understand that I'm trying to show you the equator, right? So here's the equator, the equatorial plate or the metaphase plate. And again, depending on which way, whether it is the maternal chromosomes lined up pointing to one pole or the paternal pointing to the opposite pole, we're scrambling the genetic material again, all right? So this is the major event called independent assortment. That is another way to increase genetic diversity. Okay, so it's the random way that the maternal and paternal homologs line up at that metaphase plate.
Because of the crossing over that occurs during the first division, the sister chromatids in meiosis II are not always identical. Omitting a chromosome duplication before meiosis II produces haploid gametes.
Okay, so again, we're producing haploid gametes, and he talked there again about the fact that when we're looking at that interphase between meiosis 1 and 2, there is no DNA replication. So this random orientation, here's what we're talking about when we talk about independent assortment. We're talking about the way that the tetrads just randomly line up on the metaphase plate.
During metaphase 1 of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
Okay, so I'm just going to back it up there for a second. Look at what is happening here. Each of these different cells has maternal and paternal chromosomes just randomly lining up. Here, all of mums are pointing to that pole. All of dads pointing to that pole. Here, it's the exact opposite. All the paternal dads go to that pole. Mums go to this pole. Here, we've got mom, dad, right? It's random. And so we're shuffling the genes. We've already shuffled them during our crossing over, but now this acts to increase even more the genetic diversity, so that the offspring are not going to be the same as the parents. Okay. So I'm going to just wrap it up there. I'm going to come back and talk to you about mitosis versus meiosis, just a quick summary of the two. That'll be my video number 3. And one more time to go through those important events that you need to know that increase genetic diversity. All right, signing off for now, Michelle.Ladine at ghsd75.ca. Talk later.